ठीक है सर। Give me a minute.
Hello, sir. Am I audible to you?
Hello, sir. Yes, you are audible, sir.
Okay, thank you, sir.
Thank you, sir. Okay, so good afternoon to all, our esteemed resource person and beloved faculty members and all dear participants. Myself, Pradeep Jana, Assistant Professor at Bharat Technology, will be the host for today's session. Learning is a continuous journey and excellence is crafted through consistent refinement. With that spirit, we gather again today, ready to immerse ourselves into a insightful discussion that I believe will empower us in our respective academic journeys with clarity, confidence, and purpose. On behalf of Bharat Technology, I welcome you all to the second day of our 7-day faculty development program on strengthening academic excellence faculty development program for outcome-based new B.Pharm syllabus. Now it gives me immense pleasure to introduce and welcome our today's distinguished research person, Dr. Rohit Bhatia. Sir, we are truly honored to have you with us, and we look forward to gaining deep insights from your expertise. Dr. Bhatia is the head of Department of Pharmaceutical Chemistry, Chitkara College of Pharmacy, Chitkara University, Punjab. He carries a vast experience of 12.5 years in academics, administration, and research. He has specialization in medicinal chemistry, computer-aided drug designing, and bioanalysis. His area of interest includes anti-cancer drug discovery, paper-based microfluidics. He has published more than 145 review and research articles in various international and national journals. He has published more than 10 book chapters. He has also delivered more than 20 guest lectures as a speaker on various national and international platforms. He has qualified GPAT exam for 4 times. He also guided 32 postgraduate and 6 PhD students. He has a potential as a reviewer in various international reputed journals. Dr. Bhatia was the brand ambassador of B.Pharm Science in 2029 and 2024. Dr. Bhatia also participated and presented his research work at various national and international conferences. He has been granted more than 12 patents by Government of India. Recently, he got a grant of 20 lakhs by MSME India for the development of point of care device for detection of glucose in human saliva. He has been awarded with Young Scientist Award twice at international platforms, and he also authored 6 books. In today's session, Dr. Bhatia will deliver his lecture on impact of AI-driven molecular docking in drug discovery from basics to breakthroughs. So with this introduction, I will request Sir to kindly start his today's session. Sir, the session is over to you, sir.
Thank you, Pradeep, sir, for the wonderful introduction. And I would like to pay my thanks to all the management principal, sir, of Bharat Technology Institution, Rajeshri, for inviting me for this wonderful program, that is faculty 7 days faculty development program on a very recent theme, that is strengthening academic excellence, and that is a FDP program for outcome based new B.Pharm syllabus. So it is a very good initiative, and I would like to congratulate all the faculty members that they have taken such kind of initiative and before starting the new syllabus, which is just introduced by PCI, and I think in the upcoming year or in the upcoming semester it will be implemented all over the India. So the title of my talks is molecular docking, which is a part of computer-aided drug designing, which is in the curriculum according to new syllabus. In the, I think in 8th or 7th semester it is there. So in today's talk, I will cover very basic of molecular docking, which we need to make the students of undergraduate students to make understand why docking is carried out, how it is carried out, and how we can carry out it without purchasing or without spending money on costly software. So I will introduce about a free software, that is Autodoc software, which everybody can use, faculty members can use, and various good journals are accepting the docking carried out through it. So can I share my presentation, sir?
Please, sir, please.
Okay. Is it visible, sir?
Yes, sir, it is visible. Just make it, sir, full screen.
Okay.
Thank you, sir.
It's fine now, sir.
It is fine now, sir. Done.
Slides are moving.
Yes, yes, sir.
Okay, thank you.
Oh, yes, sir. Okay.
So I have kept my talk very basic, where I will cover basics of molecular docking, and everybody knows that it is time of artificial intelligence, and molecular docking and in silico studies are already the part of this AI driven computer aided drug designing. So I will cover very basic of molecular docking so that our undergraduate students and faculty members can understand. And then I will discuss little about its applications, and I will then discuss my success stories, how we have implemented molecular docking and how successful it is in the market. So the title of my talk is From Basics to Breakthrough: Impact of AI Driven Molecular Docking in Drug Discovery. So firstly, we need to understand the process of drug discovery and drug design. So basically, drug discovery is the process through which potential new therapeutic entities are identified using a combination of computational, experimental, translational, and clinical models. So drug discovery is a vast process. It takes 10 to 15 years, where firstly we are designing our drugs using in silico techniques like molecular docking, molecular dynamics, and then we are synthesizing the molecules through suitable synthetic strategy. Then we are transforming it to animal studies, preclinical studies, and finally into clinical models, human trials, etc. So despite advances in biotechnology and understanding of biological system, drug discovery is still a lengthy, costly, difficult, and inefficient process with a high attrition rate of new therapeutic discovery. So if we talk about the success rate of drug discovery, it is very slow process. Even there is a huge advancement in designing softwares, in formulation softwares, in animal models, still it takes billions of money and years of time to launch a drug from initial state into the market. So drug design is the inventive process of finding new medications based on the knowledge of a biological target. So in the most basic sense, drug design involves the design of molecules that are complementary in shape and charge to the molecular target with which they interact and bind. So if we think like a chemist or like a drug designer, so we are taking a protein as a receptor and we are studying its structure. Then we are designing our molecule according to the structure of that particular receptor. So this is the basic process of drug discovery if we use CAD process. So what are various drug design goals? Why we are going for drug discovery and designs through these molecular modeling techniques or CAD techniques? So drug design seeks to explain effect of biological compounds on the basis of molecular interaction in terms of molecular structure or precisely the physicochemical properties of the molecule involves. Because whenever we are designing a drug, it is not just a drug receptor interaction. After drug receptor interaction, the pharmacokinetics is a very important task. Even we are successful in synthesizing a drug in a good yield, even in silico results are very good with a good docking score, or good molecular dynamics predictions. Still, if the ADME is not good, we cannot design a drug or we cannot launch a drug into the market until ADME properties are achieved. So the first target is to achieve the drug receptor interaction as well as ADME. So various processes by which the drug usually produce the pharmacological effects. So in the second step, we are studying how the drug is interacting with the body, how it is moving inside the body, how it makes any change inside the body. Then how the drugs specifically react with the protoplasm to elicit particular pharmacological response. So all the receptors are present in the protoplasm. So how our drug is interacting with that particular receptor through hydrogen bonding, through van der Waals forces, through hydrophobic interactions, through pi-pi interactions. This is also a goal of our drug design. How the drugs usually get modified or detoxicated, metabolized and eliminated by the organism. So I already told pharmacokinetics is the important part, and after pharmacokinetics, after the metabolism part, toxicokinetics is very important because the drug must be excreted from the body in the stipulated time period. Then probable relationship between biological activity and chemical structure. So if you are a medicinal chemist, then everybody has studied QSAR. Whenever we are launching a drug into the market, we are proposing a quantitative structural activity relationship between various fragments of a molecule which are contributing towards the activity of a particular molecule. So these are some goals of drug design which a medicinal chemist or a drug designer keeps in his or her mind. So what are various strategies for drug design? One is random screening of a large number of compounds in search of a lead with desired biological activities. So if we talk about in silico techniques, we are having a large databases where lakhs of compounds are already stored. So we are seeking newer and newer molecules to study their activity. Second part is whenever we are getting a lead molecule, then we are carrying out structural modification to the lead molecules to eliminate the toxic effects or to increase the therapeutic activity. Beyond this, we are using some rational drug designs, including different approaches combining artificial intelligence. So in rational drug design, we are capturing various structural features from the already existing drugs. Suppose we are going to design a new drug for Alzheimer's disease. So in rational drug design, we will seek already marketing drug or already marketed drug like suppose donepezil. So donepezil tacrine is there. So we will pick some structural features of those drugs. Then we will incorporate those structure in our design molecules and with the help of artificial intelligence, and then we will design a drug. This is known as rational drug design, where we are capturing the structural features on the basis of already existing molecules. So these are some strategies for drug design. So among these, in silico approaches are in trend from last 2 or 3 decades. So computer-aided drug design methodologies are playing an ever-increasing role in drug discovery that are critical in cost-effective identification of promising drug candidates. So in silico approaches are reducing the time period, reducing the cost, and most common advantage is that these are reducing the animal studies. These are reducing the is the biggest advantage of these in silico approaches. Although there are some reliability issues with in silico approaches, but still 50 to 60% reliability we have achieved till date. So these computational methods are relevant in limiting the use of animal models in pharmacological research for adding the rational design of novel and safe drug candidates. So with the help of in silico approaches, we are able to predict drug receptor interactions, type of interaction, how the drug is approaching the receptor, how drug is behaving inside the flexible environment of the body, how the drug is traveling, how the drug is getting absorbed, how it is getting distributed, how it is getting metabolized, and how it is excreted. And beyond this, we are also able to determine its toxicity also with the help of in silico approaches. So these are reducing our drug discovery time and saving billions of money and saving several animals from being sacrificed. So in silico approaches in drug discovery, these are basically two types of approaches. One is known as structure based drug design, another one is known as ligand based drug design. So in structure based drug design, we are already familiar with the structure of receptor. Means in structure based drug design, we have the knowledge of the structure of the receptor, and according to the structure of that particular receptor, we are designing our ligands, then we are carrying out docking, and then we can carrying out our molecular dynamic simulations, and ultimately we are able to get a lead molecule. And finally, if the lead molecule is getting properly substituted and properly investigated in preclinical studies, then we can launch a drug candidate into the market. So this is the structure based drug design. In case of ligand based drug design, we do not know the structure of the receptor. We are hypothetically creating a receptor structure which is known as a pharmacophore model, and that is produced with the help of homology modeling. So here only the ligand structure is known, means we are just knowing the structure of drug, not the receptor. We are applying QSAR approaches, and then we are carrying out ligand based virtual screening. In virtual screening, we are screening a huge library of design molecules through our homogenized, our hypothesized receptor, and then we are able to optimize a lead, and finally through preclinical studies we are able to get a drug candidate. So these two approaches are being largely adopted through in silico drug discovery. Beyond this, fragment based drug design is also there. That is also a part of structure based drug design only. So in structure based drug design, as I already told, ligands are not known. A receptor structure is only known. So we are creating a library design. We are preparing the library. Then we are selecting the receptor and preparing it through molecular docking studies. Then we are preparing a binding site where our drug will bind. Then we are carrying out docking. We are getting some docking scores. After post processing, we are carrying out ADME studies through in silico techniques, toxicity studies, and drug likeness studies. And then we are sending our drug into the preclinical models like animal modeling. This is the structure based drug design. In ligand based drug design, we do not know the structure of receptor. We just know the structure of ligands. Then we are, with the help of pharmacophore modeling or homology modeling, we are designing the structure of the receptor, and then that is known as a pseudo receptor. And then we are carrying out virtual screening of a huge library in the designed receptor, and ultimately we are able to get a suitable lead molecule. That lead molecule can be substituted with the various substituents. We can apply 2D QSAR, 3D QSAR to improve the biological activity on the on that particular lead molecule. So these are the two widely adopted approaches through in silico drug design. Now I will talk about molecular docking because it is the basic. The design part starts from molecular docking only, and it is also the part of curriculum of the B.Pharm new syllabus. So what are molecular docking? Molecular docking means we are studying how a particular drug is interacting with the receptor. As everybody knows, pharmacological response will be obtained only when a drug is properly reacting with the receptor. The drug may inhibit the receptor, drug may activate the receptor, it may act as a complete agonist, it may act as an antagonist, it may act as a reverse agonist, it may act as a partial agonist. Means there will be some kind of interaction between drug and receptor. Only then we are able to achieve the pharmacological response. So what is molecular docking? From the molecular docking is a computer based program. It is a in silico approach where we are studying the interaction between designed molecules and the receptor. Means we are creating an environment with the help of some software outside the body, and then we are predicting how our molecule is reacting with the receptor, or how it is able to antagonize or agonize the receptor with the help of molecular docking. So in the simulation process, the ligand position is estimated in a predicted or predefined binding site. So for molecular docking, a database that is protein data bank, it is already available. We are just downloading the structure of our protein. We are preparing our protein. We are preparing our molecule, and then we are carrying out docking. So docking is just to see how our drug is fitting inside the binding pocket of the receptor. We are not taking the complete receptor because complete receptor is a huge molecule. It is a macromolecule. We are finding out a very small binding pocket where our drug will actually bind. So that is known as protein preparation. So whenever we are able to find that particular pocket, then we are docking our molecule inside that particular pocket and we are getting some docking scores. This is the process of molecular docking. So in simple words, it is a computer based drug receptor interaction where we are getting activity in terms of some scores. This is known as molecular docking. So what is the perception of a protein for a drug designer or a medicinal chemist? So if you are a medicinal chemist, you do not say that protein is just a polymer of amino acids. You just need to think like a chemist. So if you think like a chemist, then you have to think that it has shape and charge and flexibility, and it's not just a gene product. It is a flexible molecule where we have to find a particular region where our drug will actually bind and it will execute a particular response. So this must be a perception of a protein for a medicinal chemist in actual words. So now I will discuss various terminology used in molecular docking. So many of you are medicinal chemistry teachers. So you will, whenever you will teach medicinal chemistry and CAD part to the students, you will definitely go through these terms. So what are these? So what are ligands? So ligands are generally small molecules that bind to a protein and may interfere with protein function. It may be a drug, it may be a functional group, or it may be a biomolecule. So ligand may be a drug, it may be a polymer, it may be a macromolecule, it may be a micromolecule, but it has a tendency to react with the protein. It may induce some changes inside the protein, it may activate it, it may inhibit it. So it is a ligand. And then another term is target or receptor. So target or receptor is a part of protein. It may be a peptide, it may be enzyme, it may be a nucleic acid to which a ligand binds, forms a complex and execute a pharmacological response. So the complex between ligand and target, it may be generally it should be reversible. So that after execution of pharmacological response, the ligand may ligand may be removed from that particular target. But if we want to kill a particular cell or organism, then the complex must be irreversible also. For example, in cancer cells, the drug may form irreversible covalent binding with the receptor, with the drug, with the receptor, so that it can kill the cancer cells. So interaction is generally reversible, but it may be irreversible also in case of cancer, or it may be in case of antibiotics. Another term is PDB. PDB stands for protein data bank. So protein data bank is generally a huge library. It is a database where 3D crystallized structures of each protein is stored in its actual configuration. It is, for example, you are working on a receptor aromatase. So if you want to know its three-dimensional structure or its crystalline structure, you can just download it freely from the protein data bank. So the website for protein data bank is www.rcsb.org. So you can download the crystalline structure of any protein from this particular PDB site or PDB database. It is freely accessible to everyone. Then algorithm. So algorithm is a process or a set of rules to be followed in calculation or other problem solving operations, especially by computers. So as I said, docking is a computer based program. It is a software program. So algorithm is a set of steps how our program will be executed. This is a protocol, how we are following that particular protocol, and how docking is carried out, how step by step docking will be carried out and getting some scores. This is known as algorithm. So algorithm varies from software to software. There are a lot of softwares to carry out docking studies who are carrying latest algorithms. some traditional algorithms are also available. Then another term is scoring function. So scoring functions are fast, approximate mathematical methods used to predict the strength of the non-covalent interaction between two molecules after they have been docked. So once we are carrying a docking, there is a complex between drug and receptor. So complex is already formed, but we want to know the what kind of activity it can produce. So scoring function is a quantitative function in terms of some digits, so that we can predict that the docking which we have carried out, it was good or not. For this we require a standard drug for comparison. Suppose you have designed some anti-cancer molecules which are inhibiting aromatase molecule. So you have carried out docking against aromatase, and you have get scored some scores, but you have to compare those scores with some marketed drugs to compare the docking, whether your design drugs are better or compatible or comparable to the already existing drugs in the market or not. So this is a function of scoring function. These are some quantitative parameters. After scoring function, sir, slides are moving or not?
Sir, it is not moving. It is stuck to slide number 12, sir.
It is on slide number 12. 1 second, sir. It is visible, sir. Slide number 13.
Yes, sir, it is visible now.
Okay. So after scoring function, another parameter is force field. So what is force field? Force field is a computational method that is used to estimate the forces between atoms within molecules and also between molecules. So whenever a drug is interacting with receptor, there is some kind of interaction between the receptor and the protein. The interaction may be hydrogen bonding. It is generally a weak interaction. It may be lipophilic interaction. It may be hydrophobic interaction. It may be a pi-pi kind of interaction. So force field is a computational program which measures that particular kind of interaction. Then binding free energy. Binding free energy is the sum of all the intermolecular interactions that is present between the ligand and the target. The more negative the estimated energy, the more effective is the ligand receptor association. So whenever a drug binds to a receptor, there is change in the energy. Yeah, some kind of release of energy. So that binding energy is the release of free energy whenever there is interaction between drug and the receptor. It is generally with a negative sign. So more the binding free energy, more it is considered that the good is the docking. More good is the docking. It is again strict. Sir, actually slides are again and again. Slide number 14 is visible, sir?
Yes, sir. Now slide number 14 is
Okay,
visible.
okay. So now most important component of molecular docking is docking score. So docking score is a scoring function used to predict the binding affinity of both ligand and target once it is docked. So once we have carried out docking, we are getting a large number of conformers. So suppose we are docking aspirin with COX. So whenever we are docking aspirin with COX, we are getting some kind of scores. We are getting 10 type of conformers which are having different scores. So we are selecting that particular conformer which is having maximum score. So docking score is the quantitative term which is explaining that our docking is the best. So if we are getting higher docking score means our docking is best. So this is the quantitative term. Another term is binding affinity. Binding affinity is another term used to find the efficiency of ligand target complex. So binding affinity, if binding affinity is good, it means the more easily and more efficiently your molecule will be docked inside the binding pocket of the receptor. Then pharmacophore. Pharmacophore, it is a basically part of a molecular structure that is responsible for a particular biological or pharmacological interaction that it undergoes. As I have already told, that we are selecting a very small part of a receptor where our drug actually binds. So that particular part is known as pharmacophore. Suppose we are having a molecule morphine. Morphine generally binds to mu receptors. So mu receptor is a very small part, and the pharmacophore, that is not a complete morphine molecule. Only 2-3 sites and 2-3 functional groups of morphine are just binding to that particular bonding. Rest of the structure is contributing to the pharmacokinetics. So pharmacophore is that particular small area where the molecule actually binds and responsible for pharmacological activity. The next term is pharmacophore model. Pharmacophore model is basically a hypothetical model where we are designing our receptor if we are carrying out ligand based drug design, where the structure of only ligand is known, the structure of receptor is not known, then we are going for a pharmacophore model. So whenever we are running a software, there is option for pharmacophore modeling also. Then homology modeling. Homology modeling is a procedure that generates a previously unknown protein structure by fitting its sequence into a known structure, given a certain level of sequence homology between target and template. Homology means similarity. If we are in a search of new receptor and we have a non-sequence of only 30% to the previous receptor, means if we know 30% of the receptor of a previous receptor, then on the basis of those 30% amino acids, we can design a new receptor. That's why it is known as homology. Mean there should be some kind of similarity, and on the basis of that some kind of similarity, we are designing new receptor on the basis of that particular small sequence only. This is known as homology modeling. Homology modeling is also carried out in case of ligand based drug design. Another term is virtual screening. So virtual screening is also known as high throughput screening. So in virtual screening, we are screening a huge library. So suppose we have designed 1000 molecules. So we can dock 1000 molecules on a single receptor in a single run. This is known as virtual screening. So it is a program which is available inside the various docking softwares. So virtual screening saves number of days of times. We can screen 1000 molecules in a single run. So this is the efficiency of virtual screening actually. Next is pose. Pose is generally it is a picture. It may be 2D, it may be 3D, which tells us about how our molecule is interacting with the different amino acids of the receptor. So in this slide, you can see in the left side, it is a 2D pose. In the right side, the greener one is it is a 3D pose. 3D pose is telling us that our compound is completely fitting inside the binding pocket of the molecule. And 2D pose is showing that how various functional groups of the molecule are interacting with different amino acids. So in this, you can see that OH is interacting with arginine 555. Second OH is interacting with ASP, it is forming actually hydrogen bonding. So pose is a picture which tells about tells us about the efficiency of our docking. It is generally a snapshot. So there is an option inside the software where we can get any pose. So after this terminology, I will now discuss about various types of dockings. So docking is generally of 2-3 types. First one is local docking. In case of local docking, we have already identified the binding pocket. Means we already know that where our drug is going to bind in the receptor. That is known as local docking. For example, if you download the three-dimensional X-ray crystallographic structure of any protein from protein data bank, then you will get sometimes a protein which is having already an internal ligand. Internal ligand means we already have a molecule which is already bound to the binding pocket of the molecules. Means we already know that where the drug will bind. The binding pocket is already known. So in this structure you can see, if we select aromatase enzyme, the internal ligand is oxymestane. Means we know that where aromatase is going to, where the our design drug is going to bind to the aromatase enzyme. This is known as local docking. Second type is global docking. It is the widely adopted type of docking. So in global docking, we are sir, the screen is visible. Sir, am I audible? Am I audible, sir? Okay,
You are
fine.
audible, you are visible.
Thank you, thank you. So in global docking, the cavity is not known. We do not know where our molecule is going to bind. So we have to create, or we have to prepare the cavity. We have to prepare a binding pocket. So there is an option inside the software which is known as protein preparation. So we have to go to that program of protein preparation. Then we will create a grid. We will create a binding pocket where actually our molecule will bind. So in this figure, you can see that ribavirin is a drug, that is a antiviral drug. So this is the prepared cavity for ribavirin. So global docking means we have to create the pocket by ourselves only with the help of software. Then if we talk about in other terms, then docking is of another two types. One is rigid docking, another one is flexible docking. So rigid docking means we are considering that our receptor is rigid and our drug is also rigid. Means we are considering that our receptor is stable at a one place and ligand is stable at one place. There is no flexibility, there is no movement of receptor and ligand. This is known as rigid docking. If the bond angles, bond lengths, and torsional angles of the components are not modified at any stage of complex generation, it is known as rigid body docking. Means we are not considering any kind of movement, any kind of rotation, any kind of movement either for the drug nor for the receptor. We are considering both the rigid. This is known as rigid docking. So till now, various programs are there which are carrying out rigid docking, but the reliability of rigid docking is comparatively less. So there is a program known as FRED, that is fast rigid exhaustive docking. OpenEye program is there, doc program is there, which are carrying out rigid docking. Rigid docking is fast, where we are not considering any bond angle, any bond length, any torsional angle. So in case of rigid docking, Firstly, we are preparing a pocket of the protein with the help of a receptor. Then we are preparing our ligand. Keep one thing in your mind that whenever we are docking our molecule inside a receptor, the molecule should be a relaxed. Its energy should be minimum. It should be in its relaxed position. So there is a program inside the software during ligand preparation that is known as energy minimization. So if we will click energy minimization step, the molecule will become relaxed. The energy will be minimized. Then in the minimized states, the receptor binds to the ligand binds to the receptor. This is known as rigid docking, and then we are getting a snapshot. We are calling it as a pose. No consideration of any angle, no consideration of any rotation, no consideration of any kind of torsion angle is there in rigid docking. We are getting a score in terms of some number. This is known as rigid docking. So this is an example of rigid docking. So in the left side, you can see the blue one is your molecule and red one is your receptor. In rigid docking, as we are taking the molecule, as such is binding to the receptor. There is no flexibility inside the receptor nor in the molecule. But in case of flexible docking, in the right part, if you can see protein, so protein is getting flexible as in comparison to the drug. You can see in the right part, all three positions, the drug is getting fitted inside the molecule. This is known as flexible docking, where our protein is flexible, where we can make our protein flexible so that our drug can fit inside it. This is known as flexible docking. So for flexible docking, some additional steps we have to carry out whenever we are running a software. So the algorithms for flexible docking are different. For example, stochastic search method is there. Genetic algorithm like like gold autodock is there. Simulating annealing method using autodock is there, where we can carry out flexible docking. So the flexible docking is the actual docking which is predicting the inner behavior. If we talk about inside of our body, when there is water molecules, the drug is not always fixed. It is keep moving inside our body, and the receptor is also in solution form. It is also keep moving. So flexible docking is the real picture of inner part of our body. So that's why it is more level as compared to rigid docking. There is some incremental construction method. In incremental construction method, we need a program that is known as flex. With the help of flex, we are starting from a small fragment, then we are combining more and more fragment until we get desired score or desired configuration for our receptor. This is known as flexible docking. So for flexible docking, different algorithms are there. We can use genetic algorithm, which is based on the principle of Darwin. Everybody knows Darwin principle of revolution. So it is based on that principle only. So I will not go deeper inside this because this is beyond the curriculum of the students. But you can tell them that genetic algorithm can be used for flexible docking. Another type is bound and unbound docking. So in bound docking, it is also known as testing docking method. Here, structure of ligand bound to receptor pocket is known, and docking is done to reproduce the conformation of ligand inside receptor cavity. Actually, bound docking is used to check the reliability or check the validation of the docking. So in the bound docking, we are taking the receptor, we are extracting the internal ligand, and we are then docking our molecule inside the previously prepared pocket, and then we are checking the docking score, whether the docking score is matching to the receptor or not. That is bound docking. It is used to validate the docking protocol. In unbound docking, it is also known as actual docking. Here we have no idea about the pocket size, pocket shape. We have to create our own pocket. It is like a global docking. Unbound docking is another name of global docking only. So these are various steps in a docking procedure. So there are two parallel steps. One is target selection, one is ligand selection. Target selection we can do on the basis of disease. If we are going for cancer, we can study various types of receptors from the literature, various kinds of targets involved in progression of cancer. Another part is ligand selection. Ligand selection where we are selecting the structure, or we are selecting the type of any kind of molecule. So this we can do through the rational drug design approach, or from random screening also. So after target selection, we have to prepare the target. Target preparation means we have to create a binding pocket inside the receptor where our drug will bind. In ligand preparation, we have to make our molecule energy minimized, mean we have to make it in the relaxed position so that it can bind efficiently with the target. Then we have to run the docking protocol. Docking protocol takes 1 to 3-4 minutes, and after that you will get some conformations and you will get some docking scores in terms of some numbers. Then you will evaluate those scores, and then you can create poses, you can case snapshots to study 2D and 3D poses of your docking protocol. So first step is target selection and preparation. So I have already told you that protein data bank is there. You can download any structure, any receptor from protein data bank. It is already updated. If you want to check a receptor which is launched 1 week ago, you can find it from protein data bank. So in the picture, you can see it is a structure of actual receptor. It is a complex polymer of amino acids. So you have to find out a very small region that is known as a pocket or binding cavity. So in target preparation, various steps we have to carry out. Select non-binding site through a co-crystallized ligand. Select the center of mass of the protein. Select the coordinate of the box center. Select amino acid residues. Molecular docking serves calculate necessary map files. Means in docking, in receptor preparation, we have to create a grid where we have to select some amino acids. We have to select some dimensions so that we can eliminate all the unnecessary parts and we can select only the binding pocket where our drug will actually bind. This is known as target preparation. So in this pose, in this picture, you can see a colorful grid. It is the actual pocket of the receptor where your molecule will bind. So this picture, this grid is prepared through the software Autodoc. Autodoc is a freely accessible software. You can download it and you can run it in your Windows also, in Linux mode also. It is freely available and it is highly accepted by all journals. If you are carrying out docking through Autodoc, your paper will be acceptable in all kind of journals. This is a prepared target through MOE software and Gold software. The greener one is through MOE, that is a 3D picture, and the gray one is Gold. There are a lot of softwares. MOE and Gold are paid, and one more is Schrodinger. Schrodinger contains several type of um, programs. It is costly. Second step is ligand selection and preparation. So for ligand selection preparation, you have to use any sketching software like ChemDraw. You will draw 2D structure of ChemDraw, 2D structure of your molecule in ChemDraw. Then you can save your ligand in any format according to softwares like MDL, Mol, Sibyl, PDB, QDB, PT, Smiles format, etc. Then you will select some parameters. You will you will delete some hydrogen molecules, you will delete some water molecules, you will energy minimize your molecule. So ultimately, when you will get a relaxed configuration of your molecule, you can select it. You will see that your molecule is prepared. So you can see in this picture, these are two molecules which are energy minimized. So you can see these are in relaxed position, their energy is minimum. So actually, why this is needed? If we are going for heterocyclic molecules, the rings are sterically hindered. Their energy is high. These feel puckered. So to relax them, we are carrying out energy minimization. So when energy is minimized, we can see that our ligand is prepared. So when our protein is prepared, cavity is prepared, and our ligand is energy minimized, then we are firstly uploading our prepared prepared cavity, prepared receptor. Then we are uploading our prepared ligand over it, and then we are giving command run docking. Run docking will take some a few seconds to some minutes. So during docking, we have to select some force field. We have to select some suitable algorithm. We have to select some dimensions. We have to select some output file formats, and then we have to run the docking command. And finally, you will get docking scores and some poses. So these are the basically 2D docking poses which are obtained from docking. So in the left side, you can see various types of interactions. The outer part is representing the receptor, which contains different type of amino acids, and righter part is telling that what kind of distance is there between molecule and the receptor. The ideal distance between um, functional group of a particular ligand and amino acid, it should be 1.5 angstrom or less than it. If the dimension is less than 1.5 angstrom, it shows that there is good interaction between the receptor and ligand. These are the 3D docking poses where 3D structure of the protein is shown and the ligand is fit inside this the 3D pose. These are some interaction poses. In interaction poses, we are able to predict the distances. Means how much distance is there between uh, if there is any hydrogen bond. So what is the length of that hydrogen bond between receptor and the ligand? This we can predict from the interaction pose. Then finally, whenever you have run a docking protocol, you have to validate your docking protocol. Means whether you have carried out a good number of steps or not. So for this, we have to calculate RMSD value, that is known as root mean square deviation value, and this value should be between 1.5 to 2 angstrom. So for docking protocol, firstly, we are extracting the internal ligand and we are performing a redocking of our design molecule inside the predetermined cavity or the co-crystallized structure. And then the software automatically gives you RMSD value. If it is less than 2 or less than 1, the protocol is considered as validated. So this is the redocking inside targets active site. So whenever you are getting right side kind of poses, means your protocol is good. If you are getting left kind of poses, means the red one is the internal ligand and blue one is your design ligand, means both are not completely fitted over each other, means there is some kind of problem in docking protocol. But in right pose, you can see that blue one is completely fitting on the red internal ligand, means RMSD value will be very less. In this case, your protocol will be validated. So these are various steps of molecular docking, from ligand preparation, protein preparation, carrying out docking, creating poses, and ultimately validating the docking protocol. So Autodock is a freely available software. Every one of you can download it. You can run it. You can create your own poses. So now I'm going to discuss with you some success stories of molecular docking. Means molecular docking is just not a hypothetical thing. It is a very reliable technique nowadays. Means the success rate of is 30 to 40%. If we are designing 100 compounds through molecular docking, we are able to succeed 30 to 40 compounds per 100 molecules. So this is the reliability of molecular docking. So some success stories I am discussing. So one is HIV-1 integrase inhibitor. So a new binding site for drug treating AIDS has been discovered recently by a research group Scames et al. Similarly, alpha-1A adrenergic receptor has been discovered and their X-ray crystallographic structure has been already launched in the PDB software, and a large number of compounds have been filtered for this with the help of homology model using gold software. Type 1 TGF beta receptor kinase, it is also being screened out through a this molecular docking approach, and it is in clinical trial nowadays. Aurora kinase A has been widely studied using a genetic algorithm to carry out sampling file scoring function. Dopamine D3 receptor has been widely studied through docking receptors, and 10 selective and potent non-compounds has been reported against D3 receptor of dopamine. Serotonin receptor 5-HT1A has been exhaustively studied recently, and 40,000 compounds using predicts computational software has been used through this. Muscarinic M3 receptor has been studied through pharmacophore modeling by a Marriott et al. Group, and they have get good success through Astra compound database. Database like Astra, Zinc, Predict, there are a lot of number are already stored. If you want to study previously stored molecule, you can use Zinc database. In Zinc database, you will get lakhs of already stored compounds which were previously designed against some another receptor. Through this Predict software, you can get through this disco process, you can select. Some cyclooxygenase 2 receptor has been reported by Dadspor et al. Group. Some human aldose reductase inhibitor has been reported using molecular dynamics approach and virtual screening. So these are some recent success stories. Now I would like to discuss some our contributions where we have designed some molecules through molecular docking. So we have carried out design and virtual screening of coumarin quinoxaline hybrids as aromatase and HER2 inhibitors, where we have clubbed coumarin and quinoxaline, and we have carried out docking studies against tyrosine kinase, that was HER2 and aromatase kinase inhibitor. So these are the most potent compounds which we have got, and we have got anti-cancer activity against anti-cancer cell line, that was 7.82 microgram per ml as compared to 2.16 microgram per ml, that was the standard drug exemestane. Against HER2, we have get more good activity against breast cancer cell line, that was almost comparable to trastuzumab. Trastuzumab is a marketed drug against breast cancer, which works by inhibiting HER2 receptor. In another studies, we have designed 3,5 substituted 2,4-thiazolidinone dione derivatives against PPR gamma receptor, and we have get good anti-diabetic activity. PPR gamma receptor is involved in type 2 diabetes. Here we have clubbed aryl substituent with thiazolidinone derivatives. We have screened them through molecular docking. We have utilized MOE software for these. In another studies, we have designed MAO-A inhibitors, which was substituted through 135 trisubstituted pyrazoline. These were also screened through MOE receptors, MOE software, and we have got good publications for it. Then we have designed 2,4 disubstituted 1,5 dibenzodiazepines as GABA A receptor inhibitors, and the antidepressant activity we have got was 60.43%, which was comparable to the standard drug that was showing 63.23% activity. So these were the benzodiazepine receptors. Then we have designed novel rhodanine 3 acetic acid derivatives as pancreatic lipase inhibitors. So orlistat is the only drug which is acting as a hypolipidemic drug in the market. So we have designed some rhodanine 3 acetic acid derivatives, and we have got good result. This study was carried out through Gold software. Likewise, we have designed some 2,5 disubstituted 4-thiazolidinone derivatives as COX-2 inhibitors, as selective COX-2 inhibitors. Then during COVID, we have carried out drug repurposing also on the latest SARS-CoV-2 virus, and remdesivir was dogged against the latest launched PDV-ID of SARS-CoV-2, and we have got good results. So these were some of our success stories, and I am continuously working in this field from last 8 years, and we are getting good results. And soon we will be able to see a lot of molecules in market. algorithm. Am I audible?
Yes, sir, you are audible now.
Okay. So I was discussing the issues with the molecular docking. First one was conformational flexibility, because we are just considering the rigid docking only. Another one is availability of only limited number of algorithms. So most number of algorithms are handled by the Schrodinger software, but it is too costly to afford. Other softwares are having some less number of algorithms, so it is also a drawback. Then prediction of interfactors and affinities. Sometimes we are generally during docking we are ignoring the other things present inside our body, like we are ignoring the presence of water, we are just deleting the water molecules, we are ignoring presence of other factors like other metals, etc., etc. So these are generally interfering and uh, questioning the reliability of the docking protocol. Then availability of specific scoring functions are also less available. And last one is lack of automated programs. Nowadays automated programs for virtual screening are available, but these are available in very costly softwares like Schrodinger only. So Schrodinger costs more than 2 lakh for annual subscription only, so it is not affordable for all others institutions. So these were some kind of issues with molecular docking. So now I would like to conclude my talk. So docking is a most promising and recent artificial intelligence approach, and everybody from UG as well as PG students must learn it, because it is an important tool for drug discovery. It is saving our money as well as time. And if any medicinal chemist is going to work in direction of this in silico molecular docking studies, so they must work towards solving these issues which I have represented with the molecular docking. And I think this presentation will be helpful to the UG students so that they can get basic insights from the what are the basic terminology, what kinds of basic docking is there, and what is the recent trends which are going with the molecular docking. So with these words, I would like to thank all the participants for their patience listening. Thank you. Thank you, everyone. So now session is open for questioning. If anybody is having any kind of query, you can ask me.
Dear participants, if you have any query, you just unmute yourself and you can ask the question directly to our resource person. Please feel free to ask questions.
You can type in the chat box also.
Yes, you can also type in the chat box.
I again want to tell you that this Autodoc software is freely available. You can download it, you can install it, and you can run docking protocol with the help of Autodoc.
Okay, so I think there is a question from participants. Sir, a participant is asking, so what is the difference between rigid and flexible docking, and which one is better for means if you are going for a student or someone is going for a docking study, so which one will you prefer?
ओके, इट इज ए वेरी गुड क्वेश्चन. मैं सिंपल वर्ड्स में अगर आपको समझाऊं, रिजिड डॉकिंग में हम मान के ये चलते हैं कि हमारा रिसेप्टर भी स्टेबल है, और जो आपका ड्रग है वो भी स्टेबल है. वो ना तो ड्रग मूव कर रही है, ना तो रिसेप्टर मूव कर रहा है सॉफ्टवेयर में. बट ऐसा बॉडी के अंदर तो नहीं है. बॉडी के अंदर ड्रग भी मूव करेगी और रिसेप्टर भी एक फ्लेक्सिबल स्टेट में रहता है. तो जो रिजिड डॉकिंग है वो हमें सिर्फ रिजिड स्टेट में रिसेप्टर और लाइगेंड को रख के की जाती है. तो जो फ्लेक्सिबल डॉकिंग है उसमें हम ऐसा एनवायरनमेंट क्रिएट करते हैं जिसमें आपका लाइगेंड भी फ्लेक्सिबल रहता है और आपका रिसेप्टर भी फ्लेक्सिबल रहता है. तो ऑब्वियसली जो फ्लेक्सिबल डॉकिंग है दैट इज बेटर वन. सो फॉर फ्लेक्सिबल डॉकिंग वी आर गोइंग फॉर मॉलिक्यूलर डायनामिक्स स्टडीज. सर यू आर म्यूट.
Okay, so another participant is asking a question related to Autodoc. Okay, he is asking
या,
what are the softwares that need to be installed for Autodoc to operate this or run this?
whenever you will search Autodoc software, you will get a tutorial. You can get a tutorial of mine also. Mine tutorial on Autodoc is also available on YouTube. Another tutorial of this Autodoc is available on YouTube. You will get a link from YouTube from where you have to download it, how you have to install it, and how you have to create the files. You will get everything on the YouTube.
Okay, so thank you so much, sir, for the resolving the queries. So on behalf of Bharat Technology, and now I would like to means appreciate your such an informative and such an elaborative session. I believe our participants has get very much knowledge about the molecular docking process, and I also thank our participants for their patience learning. Now I would request our technical team to share the certificate as a token of appreciation for our research person.
Yes, sir. It is visible on the screen. I am grateful for this. And I again want to thank Rajarshi. Rajarshi was our student. We have worked together in ICF College of Pharmacy, Moga. I am thankful to Pradeep Jana, sir, for connecting with me throughout the process. I would like to thank Principal, sir, Dr. Biplab Devnath, sir, for providing me opportunity. And I would like to congratulate the Bharat Technology Institution for carrying out this kind of FDP for our UG and PG students. So thank you everyone. Thanks a lot.
Thank you so much, sir, for your valuable time, and we appreciate your presence, and we will be very much fortunate to have you in our future endeavors. Thank you so much, sir. Once again, thank you to all the participants also. So with this, we conclude here our today's session. Okay. Thank you so much, sir.
Thank you, sir.
So tomorrow session will be at 11 a.m. Our next session will be at tomorrow 11 a.m. So I will request all the participants to join on time for tomorrow's session. Thank you so much. With this, we conclude our today's session here.